So as an hour ago, I realized that the things I've been doing lately are interesting for the first paper from the DANIO-CODE. I actually don't know much about what is the part that is the most interesting to you, so I encourage everyone to communicate as much during this presentation. I will just briefly mention what I showed to the people who were yesterday with us, and that is that I've been looking through mRNA in situ hybridization information from ZFIN database, and there we have about 5,500 publications that were annotating about 12,000 genes. I'm not going to go into this details, but when I started this, I didn't know almost anything about the zebrafish anatomy, and then I thought, okay, after I summarize this everything, I'll probably be expert for the zebrafish anatomy. And that's why I have this question which my wet lab colleagues, students, will be able to answer, and that is, which anatomical structures can you see in a zygote? Anyone? Yes, and then there are really interesting examples of misannotations. So for example, there is one publication that is showing that gene, oops, do I see anything? That the gene TP73 is expressed in the zygote, which is here state 1 in the brain, which, although I don't know nothing about anatomy, it seems like it cannot write V. Or there is another example where, again, you have something misannotated, where there is a proof that there are about 10 genes who show the expression, who show expression of gene in the gill. So, as I mentioned yesterday, I tried to create these hierarchies of anatomical terms, so it's easier to summarize them all, because as 5,500 publications used about 1,500 different anatomical terms, it might be difficult to summarize them. But now, when you look at the distributions of how many information in a ZFIN are coming from different anatomical systems, you can actually clearly see that the most of the data points are coming from the whole embryo or whole cell information, and then again, the biggest column or the biggest number of anatomical terms are coming from the nervous system. And that is just showing how zebrafish was very interesting for the neuronal development research. So now that I've mapped all these anatomical terms into the system, I was able to build a hierarchy, to cluster all the genes according to the anatomical system in which these genes show the expression. And then you get the heat maps that look something like this, where you have, oh wait, now I really need a pointer. Yeah, okay. So you here have a cluster of genes that only show the expression in the whole organism. But this is something that is problematic just because there are a few publications that were using whole organism very generally. So even if maybe gene wasn't expressed in the whole organism, the application said, okay, yeah, it was a whole organism. And then again, here you have a cluster of genes that were predominantly expressed in neuronal system together with some sensory system and so on. But after you would do a filtering to have only genes that were researched by at least 5 publications, you get this clear signal that difference between this plot and the previous plot, that now the whole organism almost doesn't show any information. So now the data is much more consistent. And I was just wondering whether this will be of somehow used to combine it then with RNA-seq data and then to get from, but I'm going to mention it a bit later on. And this is now showing about 2,000 genes that are there, and it's really hard to summarize anything. But if you would go and check, for example, all the PAX genes that are there in ZFIN's dataset, you can now see that there are like this example of duplicated PAX6 genes where now you see that the PAX6B, so one of the copies, shows the expression in the endocrine system or in pancreas, while the other one shows the lower expression. And these actually numbers are showing like, so red color represents no information in the ZFIN, while the lighter the color is, there are more publications that are showing that there is expression in it. And now here you can even see that the square here in the 6A copy of the gene is a bit lighter, which means there were more publications that were showing this expression. And actually, when you look at the papers who were analyzing these two copies, you actually see that A copy is predominantly and only expressed in a neural system, while the B copy is also expressed in pancreas. Yes, so that's why I was thinking, and I would encourage everyone to join me to see how could we so now incorporate these informations together with like RNA-seq data or chip that you will have, because I mean I found it really hard to say anything about this because I don't know which data sets will be there available, but it might be interesting to check like which if you have like RNA-seq data and you find a differential expression of a gene and then to correlate it somehow with these patterns of expression across the times and anatomical system, and then be able to predict if I have this differential expression, where will my mutations or what will be the outcome on the phenotype. Yes, so this is mostly what I wanted to show you, but there's one thing that Karsten was asking about the questions that we could, oops, I lost the screen, questions that we could ask with this, summarize. Now that we have a ZFIN, what questions? And then you were asking, okay, how much is gene expressed? Do we have chip-seq signal? And okay. And there is also there is an interesting way to look. If now that we've summarized all these anatomical terms, you can ask also questions of how many genes are expressed in, for example, this is yolk syncytial layer. How many genes are expressed there, and how does this expression change? And then this blue polygon is showing the total number of genes that were expressed in the different stages, while the yellow one shows only the genes that were expressed in the previous stages. So here you can clearly see that there were 2 time points where there were introduction of a lot of new genes that weren't expressed before. So yeah, that is also one type of questions that could be asked.